Hi, my name is Cherish Wiley, freshman biology student on North Carolina A&T, and this is my protein and mutation project. The protein that I was assigned is the type 1 collagen protein. The mutation of this protein that we're looking at is the dominant negative substitution of glycine for a larger amino acid, and the condition that this mutation causes is osteogenesis imperfecta, also known as brittle bone disease. The normal function of type 1 collagen is to form fibers that serve as the framework for bone, skin, tissues, other connective tissues throughout our body. Collagen is a structural protein that plays an important role in the body. Collagen gives our body structure by protecting and maintaining our softer tissues while attaching them to the skeleton. Type 1 collagen is normally found in our extracellular matrix. Type 1 collagen is made up of a unique combination of amino acid that does not occur naturally in plants and can only be found in animals. Type 1 collagen is most abundant in our connective tissue, such as skin, bones, and cartilage. Here's my 3D model of type 1 collagen. The pink and purple sides represent the two alpha 1 chains, and the blue represents the alpha 2 chain in type 1 collagen. These different colored beads represent glycine, and that is because glycine is a relatively small amino acid and only contains hydrogens. This is what type 1 collagen looks like when it is mutated. As you see here on the left-hand side, glycine is still present, represented by the different colored beads, and the triple helix still has its twists and curves. But as you go down here, you see that the beads are missing. There is no more glycine, and it is replaced by cysteine. Cysteine is a much larger amino acid than the glycine is, and when cysteine is present, the triple helix unwinds and it loses its structures and properties that helps form the strong fibers of collagen. When the mutation occurs, type 1 collagen becomes less active. A unique amino acid sequence is responsible for the tight triple helix of collagen. Glycine is the every third amino acid, and proline and hydroxyproline are usually the other two. Type 1 collagen is made up of two pro-A1 chains and one A2 chains. The triple helix unwinds when glycine is substituted for a larger amino acid, losing its structure and strength. The different interactions that these three amino acids can participate in is for glycine and proline. Their nonpolar and hydrophobic side chains will exude water. To be structurally strong, fibrous proteins like glycine and proline need to be nonpolar. Hydroxyproline is polar and hydrophilic. That means it loves water. Strong hydrogen bonds are formed by hydroproline, which contribute to collagen strength. Glycine, proline, and hydroxyproline are relatively small amino acids. Glycine and proline are both hydrophobic and allopathic amino acids, meaning that they contain only carbons or hydrogens. They are both neutrally charged, and they both are nonpolar. Hydroxyproline, on the other hand, is hydrophilic and polar, neutrally charged, and as well as small. Proline and hydroxyproline stay the same in this mutation, but glycine is mutated for a larger amino acid like cysteine. Cysteine is hydrophilic, neutrally charged, and is a larger amino acid. Cysteine, larger shape, inhibits the tight twist and triple helix structure. The polar and hydrophilic side chains readily form hydrogen bonds and readily dissolves into water. Cysteine replacing glycine disrupts the peptide bonds, hinders the formation of the twist in the triple helix, which leads to an insufficient matrix bone or osteosynthesis. This will prevent the formation of strong fibrous structures of collagen and causes osteogenesis imperfecta, or brittle bone disease. In my 3D model, there are a few limitations that do not accurately represent type 1 collagen. One thing is that my 3D model is not to scale. In the body, it is obviously smaller and much longer than this inside of our bodies. Two is that I only showed one portion of the mutation. In reality, the mutation will be all along the chain, as well as I didn't actively represent proline and hydrogen where they're located. 3, I only showed one form of collagen, and osteogenesis imperfecta. There are multiple types of collagens that naturally occur in our body, and there are more than one types of osteogenesis imperfecta, ranging from mildly to deadly depending on the amino acid. I chose to use cysteine, which usually occurs in a mild range, about 40% severity in the cases of brittle bone disease.